And then we need our two pieces. So we need our GFP gene, and we need our RNR3 promoter. So we'll be combining those three pieces together to make the whole RNR3 GFP today. Okay? So that's three pieces putting together. Just as a control in case things don't work, we're also going to build just a regular old GFP. And in this case, we're not going to specify the promoter, but it's going to be a promoter that works all the time. So whether or not the DNA is damaged, the cells should just be green all the time. So we're going to be combining a vector and a GFP gene. So this one, when we put it in cells, they should just be green. Any questions about that? So if you assembled a good GFP product last time and you ran on a gel and we got in a really nice band and we all oohed and awed over it, I remember that. Use your GFP for this construct. Just combine your GFP with a vector. So your GFP products are here. So combine yours from last time, and then I've given you a vector right here. Right. So you just combine those two pieces together. Okay? If you didn't, if you weren't here, or if your GFP didn't assemble really well, then go ahead and do this construct, and I've got the three different pieces. The RNR3 promoter, the GFP, and the vector, and they're all here. So you can come and take one of each of those. And that way, approximately half of us will be building this, and approximately half of us will be building that, and all together we'll get them both.
I'm sorry, what GFP stand for?
Green fluorescent protein.
I'm sorry, what is a promoter?
So the promoter for this is what we call a constitutive promoter, one that's present and is there all the time. So that's cell is going to be making GFP all the time.
So GFP, that's the marker here.
GFP is the marker gene, or the reporter, yep. Okay, any questions on that? Okay, so I told you we're going to be doing 3 assemblies. This is only 2. So we're going to make 2 constructs. Just in case one doesn't work, maybe the other one will, but we're also going to assemble them using 2 different methods. So one we're going to use what we call in vitro or in the test tube method, and that's going to be what's called Gibson assembly. Because it was developed by Dan Gibson at the Venter Institute, and if you were here for John Glass's talk, he told you a little bit about it. So Gibson assembly has kind of rapidly become kind of the standard cutting-edge method to assemble DNAs. It used to be, and probably the iGEM team and most labs are still using traditional, what we call restriction digest to put together pieces of DNA. In other words, use a restriction enzyme, and a restriction enzyme recognizes a very specific sequence of DNA and cuts at that sequence. So if I wanted to put these 2 pieces of DNA together using a restriction enzyme, I could only do that if they both had the same sequence that was recognized by the same enzyme. And you could design or engineer them so that they had the same restriction site. But there's a good bit of planning and designing and manipulating DNAs so that they have what we call compatible or the same restriction enzyme site. So it's a little bit of a drawback. So Gibson assembly has really replaced traditional restriction enzyme cloning. And the reason for that is because Gibson assembly will work regardless of what sequence you have. It doesn't have to have a specific site. All it needs is for your 2 pieces of DNA to have the same sequence at their ends. So let me tell you a little bit about how Gibson assembly works. How do I get it to find the source?
You just needed to wiggle something.
Just wiggle in a bit, that's the interesting. Okay, so here's how Gibson assembly works. You take 2 double-stranded DNA fragments with overlapping ends. So in one case, this would be our vector, which would actually be a circle, and then our insert, which would be GFP. You can do this with 2 to 3 to 4 different pieces of DNA. And then what you do is you combine your DNAs with something called Gibson assembly master mix, which You know, Dan Gibson licensed to a company, NEB, so he probably now gets 50 cents every time we set one up. NEB certainly charges me more than 50 cents for the master mix. So these are very convenient reactions, but I'll point out that they're not exactly cheap. And the reason for that is that master mix is actually a mix of 3 different enzymes all mixed together. So the first is an enzyme called an exonuclease. And the exonuclease, what it does is it takes double-stranded DNA, and we say it chews it back. It cuts the end. Okay? So you can see here it's taken this double-stranded piece of DNA, and it's cut the end and kind of left a single-stranded piece. Okay? It's really important that it's only cutting one of the 2 strands. And the reason for that is because it's chewed back and now left single-stranded DNA. Okay? Because these 2 sequences overlap or they're complementary, they can hydrogen bond to each other and start forming bonds, and these 2 different pieces of DNA can now come together.
So the 5 ends are still joined, right?
What?
The 5 ends are still joined. The 3 ends are separated out.
The 3 ends are separated out. This piece is still
The 5
base
is-
paired with its
So the 5 to 5 is still
template.
joined.
Mhm. Okay. Any other questions about that? Okay, so now they've annealed or base paired together. The second enzyme comes in, and that's a DNA polymerase. You'll notice that the exonuclease is very non-specific. I said it chews back, and it just kind of chews back randomly. It's not stopping at any particular place. It's just chewing back a bunch of nucleotides until eventually it falls off the template. So it can chew back actually a couple hundred nucleotides in either direction. All it needs to do is leave enough here for these 2 to anneal together, and then the polymerase comes in, and you can see there's this gap, this single-stranded region. So what the polymerase does is it fills in that gap. It takes any single-stranded DNA and it converts it back into double-stranded. Okay? So exonuclease chews back, DNA polymerase fills in, and then we need one last enzyme to kind of seal each of these strands so that we have a full double-stranded molecule, and that's called DNA ligase. Okay, so ligase comes in, and at the end you've got one double-stranded DNA that now has these 2 pieces joined together. Okay? So it's really easy, again, just because the exonuclease is so non-specific, it'll just chew back enough to leave what we call a sticky end, or a single-stranded overlap that can anneal together. And the really remarkable thing about this is that they all are working at the same time. You see, we're not putting them in 1 and then 2 and then 3. They're all mixed together, which means exonuclease and polymerase and ligase are all working at the same time, and it works at 50 degrees. I think this is old because now they recommend only 15 minutes. So it's a very quick set of reactions. Kind of magic in it too, that this actually works.
I have a question. Like after the exonuclease, after it chews back the ends, how does the DNA fragments anneal since the 2 pieces won't like exactly match up?
Well, they do match up. So these single-stranded, where it's red and where it's blue, are perfect matches.
Oh,
So these 2 single strands make perfect base pairs together. So it's thermodynamically favorable. It's favorable for them to bond. But you're right that they don't pair up everywhere, right? You have these little gaps, and the molecule seems to be fine having those single-stranded gaps for a while until DNA polymerase fills them in. Now again, the key is that this is a very transient state, right? That the polymerase is right there, so it's coming in and filling these in probably very quickly.
So my question would be like, let's say the ligase does the annealing of the part. What stopped the exonuclease again from chopping off the final product? The last stage.
From here, from coming back
Let's come back to
in.
let's come to the last one. Like,
Mhm. See, I hit the wrong thing.
the last one, the DNA ligase works.
Right.
What's to stop the exonuclease from again chewing up?
Come here and chewing up.
Wait, this one also.
Oh, well, exonuclease will only work at the end of a DNA molecule. It can't come into a fully closed DNA molecule like this one,
Mm-hmm.
it needs an N to jump onto. So exo, the prefix means ends. So there are some nucleases that are endo that can come in the middle, but exo can only come in on a double-stranded end.
How does it only choose one side?
Of
Like-
only this end and not that end as well.
Yeah.
So it's just picking which exonuclease you use. So we have actually a whole bunch of different, so nuclease means an enzyme that cuts DNA, nucleic acid.
Okay.
This one actually is T5 exonuclease, meaning that it was isolated from the T5 bacteriophage, which is a virus. And it has been characterized in the lab and known to have this activity. You could find a different exonuclease that would chew this end instead of that one. So it's just that Dan Gibson happened to probably try a whole bunch, and he picked this one as the one that worked.
Okay.
But the enzyme has only that one activity.
So you could presumably use a different exonuclease to, in the same sort of mix, and come up with something that he hadn't patented?
You can have
You
three.
could make your own Gibson assembly mix, yes. And people do kind of homemade this, and you can buy each enzyme individually instead of buying master mix and pay a lot less for them individually and make them work, yes.
So what's the
The
advantage?
same principle would work with other enzymes.
So what's the advantage of using the master mix when you can buy them separately?
The advantage of using the master mix is that you do get all three enzymes in the correct ratio.
All the ratios.
And the master mix is not just the enzymes, but it's also a buffer. And there's actually some cofactors that these three enzymes would like to have around. I did look into buying them individually and just making a whole big pot for bugs of master mix that we could use. But actually, once you buy all the components and those specialized cofactors, it winds up being kind of just as much, and then they're not as optimized. So, you know, when people home make it, things don't work quite as well. You know, they charge you money because it does work.
So it seems like the more the ends are chewed back, the less likely, just statistically, it's going to be it's going to find a compatible end to bond with. What stops it from bonding early? Let's say after a few base pairs have been chewed back, it's going to find more things that maybe you don't want it to bond with.
Right, but the bonds that it makes are also going to be less stable. So if you
Right.
only have 4 nucleotides that overlap each other, it's going to be relatively weak bonds. So
Right.
at 50 degrees, the bonds are constantly being made and reformed. So if
Okay.
they're not very stable, there's not a lot of likelihood that it's going to survive this extension step.
Got it. Okay.
But you're right that it's not 100%. And in
Right.
fact, some of my friends who have used it say that there is some error, especially in this step of filling in,
Mhm.
that there are some errors that get incorporated.
Okay.
So just like anything else, it's good, but it's not perfect.
How many pairs of overlap do you need?
Generally, they recommend 40 to 80 base pairs of overlap. Yeah. Oh no, wait, I'm sorry, that's for yeast assemblies, we're gonna do it next. For Gibson, it's less. I think they want, maybe it's 15 to 40. So it's not a whole lot.
I'm sorry, what's A stand for, B for,
Oh, just fragment A, fragment B,
all four,
which
right?
would be for us our vector and our GFP gene, just the 2 different pieces.
So, so why we need or why we have too much, what is, why we need to add
Uh-huh.
additional,
Why do we need the
Control.
RNR3
Yeah.
and the GFP?
yeah, what's the two different, what's the difference between the two, why we need it?
Well, so this one's just going to express GFP at all times and in all places. This one's going to express GFP only under certain conditions, only when the RNR3 allows it to be expressed, which is under conditions of DNA damage. So these two should act differently in itself.
And how is it that what we've put together does have those overlapping? Did you engineer that in?
Oh, yeah.
Did the synthesis?
So you can go into the NEB site. The other nice reason to buy from them is, okay, really so inept. The other nice reason to buy from them is they have a website called NE Builder. And you put in your DNA fragment, so you input this sequence, you input that sequence, and it tells you how to design your fragments. So in other words, if this is our GFP gene, our vector, I designed it by PCR, I amplified, and I put on ends that are complementary, that overlap GFP.
Okay.
So yes.
So you modified that vector specifically?
I modified the vector so it's compatible with
Right.
GFP. Yep. And then this vector, so you'll notice that for people who are doing GFP, you have one vector. For people who are doing the RNR3 GFP, you have something called labeled RNR3 vector. And the reason is this vector overlaps GFP at both ends. This vector overlaps GFP at one end, it overlaps RNR3 at the other end. So almost all of the vector is the same, except the ends are different between this one and this one, because they have different overlaps.
And then was the GFP engineered to match up to the RNR?
Yeah, this GFP I had to redesign the end. No, I'm sorry, the RNR3 I engineered so it would
Okay.
overlap. So the GFP is constant, I just engineered what it overlaps with. So it would be.
So GFP comes from the biobrick.
Comes with the BioBrick?
You get it from there, right? No.
No, we assembled that from
Everything?
small pieces from all of this. Yeah. So that's synthetic. So the GFP is synthetic.
The vector standard.
The vector is standard, but again, I created it to have ends that were compatible.
So you designed the software that we talked to us about last time. The one where you design the genes and
The gene design?
gene design.
Mhm.
Design. That's where you design all this.
So gene design is where you can design the synthetic gene,
Mm-hmm.
and then any builder is where you design your vector and
And
your
your
promoter so that they'll all go together
And then you
properly
send
and
it-
have those 15 to 40 base pair overlaps.
And then you send it to the company to make it for you.
So I bought all of those that allow me to amplify the vector and create the ends. Yes.
And is there a reason why that's preferred instead of just when we synthesize the GFP,
Uh-huh.
just adding those tails to match the vector?
Oh, you could do it either way, right? So you could use a vector that you had in the freezer
Mhm.
and then design GFP so it overlapped, or you could use the GFP and make the vector overlap. So either one, just depends on where you started with. Okay, good questions? Okay, so that's the first two things we're gonna do is Gibson assembly, and you'll see it's super easy. All you're gonna do, I think the protocols on your bench is you're mixing together the promoter. If you're doing synthetic GFP instead of promoter, just add water. The vector, the GFP gene, 3 microliters of each, and then the Gibson assembly master mix, which is right here. You've got 1 tube of it. Like I said, it is kind of expensive, so we have just enough for everybody to do theirs. But you're gonna add 10 microliters. You just mix it up and down with a pipette, up and down a couple times. You put it at 50 degrees, which is right here, for 15 minutes. Those 3 reactions are all happening magically. And then we're just gonna store it at -20, and then in 2 weeks we'll put our construct into bacteria. And if you have this one, the bacteria should glow green. If you have this one, we'll have to wait and screen to make sure that you actually have the construct. Because this one isn't gonna glow green because there's no DNA damage. If you have a GFP that you assembled well, that you've got a nice, beautiful band on the gel, do this one. If you don't, do that. Once we've done that, and that shouldn't take too long, the third way that we're going to assemble, we're just making same thing, this construct, but instead of using Gibson assembly, we're going to use yeast assembly. And I kind of wanted you to see both. So Gibson assembly is really awesome. It works well if you've got 2 pieces. It'll work beautifully. If you have 3 pieces of DNA, it'll join them together pretty well. It says you can do 4 pieces, maybe 5. Once you get more pieces of DNA that you want to put together, you've really reached the limit of what Gibson assembly can do. So instead what we do is we assemble genes into yeast. So anytime we have really multiple fragments of DNA that we want to put together, yeast will do that for us. In yeast, you don't actually need enzymes. Yeast has a terrific property called recombination. It exists in all cells. It just works at a much higher rate in yeast. Yeast love to do this process of recombination. It's a natural process of breaking and rejoining DNA. And just like Gibson assembly, it takes any pieces of DNA that overlaps and it joins them together. So we're going to take our 3 pieces, put them together, insert them into a yeast cell, and the yeast will combine them all into one closed DNA circle by magic. And yeast assembly is much more efficient. There's reports of doing, up to 25 different pieces of DNA getting put together into one construct in yeast cells. So when we talk about like building genomes, so the Venter group, which built the bacterial genome, put small pieces of DNA together by Gibson assembly. Once they wanted to really build something the size of chromosomes, they moved into yeast. So when we're talking about building big things like genomes, it's usually a combination of in vitro assembly for small pieces, but then moving into yeast when you really want to build a full, larger genome and when you want to assemble more pieces.
So why can't we do the small things in yeast too? Or is there advantage using the Gibson assembly?
You could. First of all, a lot of people aren't familiar with working with yeast, so they don't do it because they don't do it. It requires special media and not a lot of expertise, but a little bit. The other difference is, so when we do this, you're actually going to get different pieces compared to this one. And the reason is yeast love to do recombination, but they need a slightly longer overlap. So whereas the overlaps for Gibson assembly are only maybe 15 to 40, for yeast they like more like 40 to 60 base pairs of overlap.
The reason I ask is because Venture has people who are skilled. They would be able to work in either field, yeast or with Gibson.
Right.
So why would they choose?
Oh, why would they choose Gibson?
Because that's more
well,
expensive.
Gibson is 15 minutes, right?
Well, that's the reason why. Well, this
And
is
yeast
25.
is 2 days.
2 days.
Yeah. Because the yeast cells have to grow. So the yeast pick up the DNA, and then they have to grow.
Yeah, what if
So it's a little bit longer as well. Then do people like things fast?
they had a race to win
Yeah.
with an IH.
Okay, so we'll do Gibson assembly first, and then I've got the yeast stuff here. So if you're doing just GFP, if you like your product, your PCR product and the vector are here. If you want to do the RNR3 GFP, things are here. The master mix, like I said, is up on the bench. So once you've got your DNAs mixed together, you're going to get the master mix.
Sure. I think you're doing the second one though, right?
Yeah.
The first.
That's these steps here.
Since one didn't work out.
You got 10. I think the
That's a very, so if you have a double-stranded DNA here, so PCR assembly, what happens is it's a 3-step procedure. The first, you're going to denature, so you heat up to about 95 degrees, you separate the 2 DNA strands, then you put it on your PCR primer, and you want 1 amino to each strand. So this one is going to base pair here, and that one base pair there. So step 2 is what we call annealing, and that's between 45 to 60 degrees C. So that allows those 2 to base pair. And the third step is what we call extension, which is when you use Taq polymerase, which is the enzyme. And then TAC takes this and extends it all the way out. So you have a full double helix. This one is a double helix. So you go from one starting double helix to two, and then you just repeat that over and over. You get exponential. So these primers have to anneal to your sequence that you want to amplify, but at the end, it can have overlap, right, to another fragment. You can add as much overlap on those ends as you want.
And what is the have to have that primer for have to start adding?
Because that's simply because if you just give it a single-stranded piece of DNA tack, any DNA polymerase has no place to start. It needs, at the end of there's actually a chemical group, a hydroxyl group, on the third carbon, 3' hydroxyl group, and every DNA polymerase needs that chemical group to start synthesis.
So it always builds from the
It always builds from, it always needs some place to start. Yeah. Feel good? 2 more people?
These are the big ones.
Okay, so just to kind of remind everybody about like what we're doing. So right now we just did Gibson assembly, so our 3 pieces or 2 pieces should all be joined in one circle, but again, just in a test tube. We're going to put our 3 pieces together in a yeast cell, and the yeast then will join those 3 pieces together into one. So this is, again, in vitro, just plain DNA in a test tube. This is actually going to be in a cell. In session 4, we're going to take this piece out of yeast, so you'll actually learn how to extract DNA back out of yeast. So extract plasmid, and then we'll put them into bacteria. Both the one that we are going to make in yeast right now and this one that you just built in a test tube. So put into bacteria the Gibson, and we'll put into bacteria the one made in yeast. Okay? If we wanted to, right now, our Gibson assembly reactions are ready to put into bacteria. We're just waiting so we catch the yeast guys up, and we'll do it all at once.
Yeah, you'll have to ask her for.
And then in session 5, we screen. We actually look at those bacteria and we ask, do we have what we want? Do we have our GFP gene in there, and is it intact? And we'll screen by PCR. So we'll talk about PCR a little bit more then. So that's just kind of the overview of what we're doing. Yeah? Okay, so the yeast are ready to go. We've got new DNA fragments up front because, again, it's the same pieces. The sequence is the same. The only thing that's different is that the overlaps are longer for these pieces of DNA. So you have, on ice, you're going to want to come up and get, it's going to be called yeast vector, RNR3. Now it's marked with a Y, just to tell you that it's for yeast assembly, and GFP for yeast assembly. So those 3 pieces of DNA you're going to need. Okay. And then the yeast cells are right up here. So the first thing that you need to do is make a master mix for the yeast transformation. So in other words, to encourage the yeast to take up DNA, they need a couple other chemicals. The first is PEG, which is polyethylene glycol. When you work with it, you'll notice it's really, really super viscous. And the reason for the PEG is that, you know, you've got a yeast cell, which has a lot of cytoplasm in there, right? You're putting in the DNA, you want the DNA, our goal is to get the DNA into the yeast cell, right? The 3 pieces of DNA that we're trying to put in. PEG is really viscous, and it just helps the double-stranded DNA to kind of stick to the outside of the yeast cell. So it helps DNA to adhere to the outside of the cell. In other words, it just concentrates it all around the yeast cell wall so that it can get in. There's lithium acetate. We don't really know how lithium acetate works in yeast transformation, but it seems to be really important. The best guess of how it works is that it neutralizes the yeast cell wall slightly positively charged. The best guess is that it helps to neutralize the charges on the yeast cell wall so that the DNA can get close to the cell again.
It works on a cell membrane? It works on a cell membrane by transforming it?
The cell wall.
Cell wall.
Yeah. So yeast have both the cell membrane and a thick cell wall. And then the last thing, the most fun, is that even though we're putting in a lot of DNA, the DNA that we're putting in is actually fairly dilute. We're not giving it a whole lot. So DNA seems to get into the yeast cell more efficiently when there's more of it. So all we're doing is you're adding your 3 DNAs, plus you're adding an excess of extra junk DNA, just to kind of, we call it carrier DNA. It helps carry your DNA into the cell. So it's single-stranded DNA, so it's really important that the DNA is denatured. Instead of a double helix, it's been melted or heated up to separate the 2 strands. So that's the only thing that's really important that you keep on ice. Because the 2 strands have been separated, if you allow them to come to room temperature, they're going to come together again. So make sure the single-stranded DNA, it's called SS, is kept on ice. And you'll notice from the directions, it's herring sperm DNA. Sometimes we use salmon sperm or herring sperm, just because, you know, fishermen cut up the fish and there's a ton of cells right there and all that sperm, and it's just an easy source of DNA. So, and it makes undergraduates kind of all look at each other and go, ah! So it's fun that way. So the PEG, because it's really viscous, I already put exactly how much you need in each tube, which is 960. So all you're doing is, from the lithium acetate, take 144 of that, add it into the PEG, add the single-stranded DNA into that, and add some water, mix it up. Again, you're going to want to vortex in step 1 pretty well because the PEG is viscous, so just to make sure it's well distributed. Any questions about that?
What does the water do? I mean, because,
What does the water do?
because you already have the P
No,
PEG, which is very
it just keeps the concentration of DNA.
That's all.
Exactly right, yeah. Any questions about that? Now, the one thing that I did not get to do this morning was to distribute out the yeast cells, so they're right there, into individual tubes. So for step 2, right before you do that, when it says obtain 3 tubes of yeast competent cells, you're just going to come to this tube and take 3 tubes and put 100 microliters of yeast cells into each of your tubes. That's all of it is. So you're going to have 3. 1 is going to be your assembly reaction. 1 is going to be a negative control, so just water. And then the 3rd is going to be positive control. So this is a DNA that's already in one piece. It should go into the yeast cells really nicely and really easily. It's just a control to make sure that all the steps are good. So those are labeled with a plus. That's your plus. Okay? Any questions about that? So you're going to have 3 tubes. You add your yeast 100 microliters into each of them. That's step 2. You spin them down in a centrifuge for 1 minute. You get rid of all the liquid. Just toss it out. Then you add 286 of that master mix that you made in step 1. Again, you're going to want to pipette up and down a couple times because that PEG is going to be really viscous. And then you add the DNA. So in the tube that's labeled GFP, you're going to add the yeast vector, you're going to add the RNR3, you're going to add the GFP. In your negative control, you just add water. In your positive control, you add from the tube that's labeled plus. Okay? Any questions about that? Just vortex it a little bit to mix up the DNA and the yeast cells, and then you do what we call a heat shock step. So if you've done bacterial transformation, we'll do them next week, you know that we also heat shock bacteria. It tends to permeabilize the cell membrane. So in other words, it makes it more porous, which allows the DNA to get in. If you've done it with bacteria, you know that the heat shock step with bacteria is about 30 seconds. So you heat up the cell very quickly, make the membrane kind of permeable, and allow the DNA to get in. In bacteria, it's very quick. In yeast cells, you can see in step 10, we actually do it for 20 minutes. And that's again because yeast have that really thick cell wall. So we're doing the exact same thing, you just need to do it for a lot more time to allow the DNA to get in past that really thick cell wall. Okay? Once that's done, the DNA is now in your yeast cells. Essentially all you do is you spin down your cells, you remove all the liquid, you resuspend it in water, and then you put it on a plate. And I'll show you how to plate them when the time comes. And then you allow the yeast cells to grow. Any questions? So the reason for plating, I should say there's one more difference between the vector in the yeast assembly and the others. The yeast vector has a gene called His3. His3 is a yeast gene that's involved in making histidine, which is one of the amino acids that yeast need to live. So the starting strain right here cannot make its own histidine. These plates don't have any histidine on them. So if I just take these cells and put them on the plates without any histidine, they're not going to grow. So your negative control should not grow because it can't make histidine. Your positive control is a plasmid that contains the His3 gene, which lets them make histidine. So when you put your positive control in, the yeast can make histidine, the yeast can grow. Same thing with this vector. The vector contains the His3 gene. So if all three pieces of your DNA come together, you now have an intact His3 gene, and the yeast cells should be able to grow, and they'll make nice little colonies. It takes them about 2 days to grow up. But that's how you can tell that your three pieces have come together, because you'll get colonies. We say that they're His plus colonies, because they can make their own histidine and grow without on plates lacking histidine. Any questions about that? Okay, so again, place to start is to make that transformation mix. The PEG and the lithium acetate is here. Single strand of DNA is on ice. So everything's either on this tube or on the ice.